Mitosis and meiosis. Mitosis is the simpler of two processes and is carried out by non-reproductive somatic cells. Cells are tasked with the project of creating two identical cells, known as daughter cells. The first step of mitosis is prophase, in which chromosomes are duplicated and held together at centromeres. They are referred to as sister chromatids. In prometaphase, the nuclear envelope disappears and spindle fibers develop and attach to either side of the sister chromatid. The next stage, metaphase, is the stage in which sister chromatids line up down the center of the cell, still attached to either side of the spindle. It is in anaphase that the cells begin to elongate, with the spindle fibers shortening, separating the sister chromatids from one another. In telophase and cytokinesis, often paired together, the spindle fibers dissociate from one another. The cell cleaves into two cells, known as the daughter cells, and the nuclear envelope reforms. Mitosis is now complete. The two daughter cells are identical to the parent cell. Meiosis is very similar, but undergoes the process of mitosis twice with some alterations. The differing process results from the cell's need to create gametes as opposed to identical somatic cells. This means that the ploidy state in the parent cell is 2N, but the daughter cells need only just 1, or just N. In prophase 1, the chromosomes duplicate as they did in mitosis and pair up with their homologous chromosome, forming a homologous chromosome pair. These joined chromosomes then experience chromosomal crossing over, a process in which genetic information is swapped with the homologous chromosomes. This increases genetic variation and potential offspring. In prometaphase 1, not shown here, spindle fibers form and connect to either side of the chromosomal pair, as they did in the sister chromatids of mitosis. In metaphase 1, they align down the center. Spindle fibers pull apart the chromosomes in anaphase 1, and in telophase and cytokinesis, they create two non-identical daughter cells. However, their ploidy state is still 2N. These two cells then undergo prophase 2, in which the nuclear envelope begins to break down. In metaphase 2, spindle fibers form and attach to either side of the now sister chromatids as they did in mitosis. In anaphase 2, telophase 2, and cytokinesis 2, the same process is followed as in mitosis. The second round of meiosis produces 4 non-identical daughter cells, each with a ploidy state of N. These cells are gametes and continue on to fertilization.